Okay, other environmental factors that are going to come into play. It's going to be, well, what other microbes, what other cells are in the environment? People not in science have taken the word symbiosis, symbiotic, and they have taken it and redefined it to mean that two things working together. No, technically that's not what symbiosis is. Symbiosis just means two or more organisms in a close partnership. Done. Whether that partnership means that both of them gain, only one of them gain, one of them doesn't even know the other's there, well, that's why we see symbiosis is broken up into the 5 categories. When you look at the 5 categories here, symbiotic, non-symbiotic. Symbiotic, 2 organisms, close relationship. Relationship can be good, bad, indifferent. If it's good, that's mutualism. That means both are going to gain something out of it. Both will benefit. If it's commensal, one gets nothing out of it. The other benefits. But the first one that doesn't get any benefit also doesn't get hurt. Many of the microbes we have in our gut don't do a thing for us. They're just there, taking up space, and that's it. And then there's the parasites, parasitism. One organism benefits at the cost of another. These are the microbes that infect us, that cause us harm, things that we think of as pathogen. Then you also get what's referred to as non-symbiotic. This relationship, these organisms are in close proximity. We also have to look at non-symbiotic relationships. Organisms are free-living relationships not required for survival. Okay? Free living means that every cell for itself. The synergism, means that there will be some cooperation between these free-living organisms. Think kelp. Kelp is an algae. They're algal cells that clump together. Still every cell for itself, but there's some sharing. They are trying to work together to an extent. And then there's antagonistic. The mere presence of one interferes with another. They compete too much. Great example of all of this, the synergy, biofilms. Biofilms are layers and layers of microbes. You have an initial bacteria or microbe of some sort, lands on a surface. No other microbe can stick, but this one, it sticks. Thanks to cellular division, 1 becomes 2, and so forth. And they start to spread. Because they're there, other cell types can now start attaching to them, adhering to them. And it just starts building up more and more and more. The cells in a biofilm will start to interact with each other via what's known as quorum sensing. They're not working together, but it's compounds that are given off in metabolic waste or compounds that are produced just to put out. That as that concentration increases, it will cause genes to turn on and turn off in other microbes, other cells, same species, different species, so that you can start to get some interplay between the microbes in a bacteria, in a biofilm. But do keep in mind, when it comes right down to it, it's every cell for itself. They're not interacting like cells in a tissue would. They are still doing whatever it takes to benefit the cell itself and nothing else.